understanding the numbers. There's a lot of numbers in these lessons, and numbers are actually pretty good for answers to multiple choice questions, so you should pay attention to that. So the first number you want to know is 1. As I mentioned earlier, 1 chromosome is 1 large DNA molecule. I think I said 1 DNA molecule equals 1 chromosome. I hope you understand they're the same thing. Second concept here is a new concept we haven't introduced, the concept of what a gene is. G-E-N-E, a gene. A gene is a specific sequence of nucleotides. So remember, the nucleotides are the A's, T's, C's, and G's. Okay? Now, since we have a double helix and we know exactly what the complementary nucleotide is, right? If this is an A, we know the complement is T. If this is a T, we know the complement is A. So we don't give the double helix. So when we talk about gene, even though it exists, right? When we talk about a gene, we talk about a single strand of nucleotides like this. A's, T's, C's, and G's. That's it. And when scientists say, or you hear in the news that scientists have identified the gene for disease X today, that means they know which chromosome, they figured out which chromosome it was on, where it started, and where it stopped. The full complement of A's, T's, C's, and G's. It's pretty phenomenal. And so we see this down here. We see this big strand of DNA, one big chromosome, right? One strand of DNA. And scientists can identify that, hey, this is where gene 1 starts, and this is where it ends. And this is so-called junk DNA, which is an interesting name. People think it doesn't do anything. Probably means it does a lot. Anyway, here's gene 2, right? Here's dead zone, non-coding as they call it, DNA. And here's another gene here. So this whole squiggly line is just one big strand of A's, T's, C's, and G's. So when people have identified a gene, all they're saying is we know where it starts and we know where it stops. We know what it does. Okay? So given the fact that you have this amount of DNA, and 1 chromosome equals 1 DNA molecule, and you have 46 individual chromosomes or 23 pairs, then we can calculate we have about 1,000 to 2,000 genes per chromosome because we have about 25,000 to 30,000 genes in the entire human genome. And the entire human genome is a phrase that means every single A, T, C, and G from every one of your chromosomes. If we can identify your entire human genome, and that's what the Human Genome Project was, and it did it over, God, I guess it's been 8 to 10 years now, right? And personalized medicine is moving forward that everyone gets a full genomic screen. It's still too expensive to do. The goal is to get a genomic screen of each individual to be less than $1,000 a pop. That's what the technotypes are doing in the scientific community. But that's the idea, and especially within this COVID crisis, people are trying to do complete genomic analysis to see if they can identify strengths or weaknesses, kind of determine why some people get really sick from COVID and why some people don't. Okay, so the human genome is your entire complement of A's, T's, C's, and G's. And obviously yours is a little different than mine. Yours is even a little different from your sisters or your brothers or your moms or your dads. Okay, and that's the secret to the future of medicine, to see if we can identify these genes and see if we can provide better care. Okay? Anyway, that's the numbers for DNA and chromosomes.